as well as proteins. And the membrane lipid, they act as a barrier, so they do not allow the hydrophilic molecule to enter inside the cell, whereas the hydrophobic molecule at a low concentration, they can enter inside the cell. Now, looking at all these, previous background, we will be talking about how the molecules, they are transported across the membrane. What are the, different mechanism by which they can enter? Is there any energetics involved? So today's the learning objective will be that you will be learning the transport process, how a molecule which is present outside the cell can enter inside the cell. Not only the chemical molecule, it is also pertinent to the ions. So how the ions they enter. So you will be learning how the charged molecule or uncharged molecule, they transfer themselves across the membrane. Now, before we move on to the membrane, that how the transport occurs, some of the idea is very important. One of them is the membrane potential. So membrane potential is a potential when ion of the opposite charge are separated by permeable membrane, and there is a transmembrane electrical gradient which is termed as a membrane potential. If you recall, we have discussed about this, that all the plasma membrane, they have a membrane potential which is a negative. Now, coming to the the way the transport can occur, that these things we will describe. But let us first go give you a general outline. What are the ways in which molecules can enter? Each one I will elaborate at the later stage. Will give you more idea. But here we are going to discuss the different types of mechanism. One of the simplest process which you have learned so far is a diffusion. So there is a simple diffusion which can occur. So whenever the diffusion occurs, this is mostly non-polar compound, and it happens down the concentration gradient. So diffusion is always a process where you have on one side very high concentration and other side low concentration. Things are moving from high concentration to low concentration, and once the concentration gradient is there is a no gradient, you will not see the transport. So simple diffusion process occur in the membrane. So the question is, the question here is that which type of molecule they are transported across the membrane by simple diffusion. Answer is simple, that non-polar compounds, they are the one which are moving across the gradient. Then comes another class or category of diffusion which is called as a facilitated diffusion. Do you understand what facilitated means? Facilitated means it is helped by something. Now, how the help occurs here is by the membrane protein. So there is a membrane protein which is helping. So here in the first process, when the molecule is entering across the cell, you know, because of the gradient. Now, this facilitated diffusion occurs down the electrochemical gradient. Now, when I say electrochemical gradient, there are two components here. One is an electro and other is a chemical. That means there is a gradient of the mass as well as a gradient of a charge. So with these two gradient, a molecule can enter from outside to inside. And during this process, the help is given by a structure. What is this structure? So you have seen here, these are lipids. What is this molecule? This
It
and
is
this?
the protein molecule.
Protein molecules. Okay, and they will be called as the transporters. So what happens that here, the molecule which is present outside, they are entering inside with the help of electrochemical gradient. It is the gradient created by the chemical, that is the mass, when the molecule or the compound which is transported is an uncharged one. As soon as it becomes charged, that means both the mass gradient and the charge gradient starts working. So we will finally find out how this can be calculated, what formula is being used. So you will learn all the basic principle of calculating the transport energy, how the energy is being generated, or from where the energy is being extracted, all those points you will learn. Now, point here is that in this case, we are not involving the energy as per se, because no energy is being involved here, except that the energy which is coming from electrochemical gradient. Is it because of the charge, because of the mass? Now, there is another transport which is called as active transport. Now, this active transport is where really the energy is involved during the electrochemical gradient process. That means some energy like ATP is being consumed. So suppose you have a molecule which is present outside. If it has to enter inside, during this process, there is expenditure of energy. ATP will be hydrolyzed, converted into ADP and PI, and the energy generated is helping the molecule to move in. This energy could be in the form of the phosphorylation, could be in another form, but yes, the transport is accompanied by the hydrolysis of ATP, and that is why this is called as an active process. When I say active process, that means what else? Here, what is the difference between this one and this one? In the first one, there is a no charge, there is a no energy involved when you are doing facilitated diffusion. Here, it is the charge, sorry, it is the energy which is involved. So in these two cases, the difference is of the energy. And that is why this process can be called as a passive process, where this is the process which is active. Now, in the passive process, you have seen that it is also possible that the protein is involved. Here, protein is involved, but you also involve energy. So, in these two differences, that in one case energy is involved, in another case energy is not involved. Now, what else could be the difference? Reading on these details, can you tell me what other difference could be? Now, in this case, you see is that any molecule or any compound or any charge is moving across the membrane down the electrochemical gradient. Okay, so suppose here, less, so here you have a more charge or more concentration gradient that enters inside, and it equilibrates the charge or chemical composition across the membrane. But here, when I am talking about active transport, the major important difference between these two is that S, which is present out, is moving inside against the concentration gradient. So these two are very important. Whenever there is an active process, it is against the concentration gradient. When it is a facilitated diffusion, it is down the electrochemical gradient. Now, this kind of active process could be secondary. So one thing is that there is only one solute which is involved. Now, secondary active transport is again against the concentration gradient, but there are two things are involved. For example, S moves from outside, it creates a gradient, and because of the gradient, when S is moving in, your ions are also moving in. So there are a couple of examples where a transport is occurring, where it becomes secondary active transport. Now, next type of the transport is through ion channel. So ion channel, it is a down the gradient. Now ion moves from one side to another side. The gate has to be opened, and once the gate is opened, it enters in. So how gate opens? Somebody is protecting it. Suppose there is a receptor here. So X molecule or ligand will come, bind to this receptor, and when it gets a signal, the ion channel opens and goes in. Another way or mechanism could be that there is an ionophore which creates a hole in the membrane. It does not require any protein or anything. It moves from outside to inside, and then it can come out. So ionophore, the movement is on both the sides. So ionophore mediated ion transport, it is a down the electrochemical gradient way it is on. Mechanism. I will elaborate them one by one with some basic understanding. So first part is that membrane potential can be created, and this membrane potential is because of the ion. So suppose I have a cell, suppose I have a cell where the gradient across the membrane for the charge is there. So suppose you have a cell, and in a cell there is a this is a cell, and here you have high concentration of the potassium ion, and here there is a low concentration of the potassium ion. So from high concentration to low concentration it will move, or vice versa. If that happens because of the membrane potential and it is along the concentration gradient, we call them as a passive transport. But suppose potassium ion enters inside the cell against the concentration gradient, what should we call them? We should call them as a active process. So is it clear for you that what is the active and passive process? I'm just going to repeat it again when we are talking about these two types. Okay, so electrochemical gradient, it is a direction in which charged solute tends to move spontaneously across the membrane, and it depends both on the chemical gradient. Suppose your potassium ion is moving, so potassium, some mass is there and charge is there. So, and the difference in the solute concentration, so if the potassium ion concentration is different across the membrane, then it is taken into account as a chemical gradient, and there will be electrical gradient because of the charge. So there is a one charge, one positive charge that will allow the gradient. Together, these two factors are referred as an electrochemical gradient or and they will create electrochemical potential. So because of this gradient, this potential is being generated. Now, looking at the transporter, if I want to classify this transporter, they are either passive transporter. So what are the transporter? What is the nature of these transporter? Any one of you? What are the transporter? Transporters are protein molecule. They are the one which are specifically involved in the transport process, and they are the one which are protein in nature. So these transporters, they are of two general types. One is a passive transporter. When I say passive transporter, that means there is no protein. Sorry, when I say passive transporter, that means no energy is involved. So I'm different active, it is basically the energy involvement. So if there is a no energy involvement, simply the molecules, they are moving across the membrane down the concentration gradient, then it is called as a passive transport or facilitated diffusion. Why we'll call facilitated diffusion? Because the diffusion is occurring, but it is also held by the protein molecule. So that is called as a passive transporter. The active transporter, they are also called as a pumps, and they can move substrate across the membrane against concentration. So remember, when I am talking about the active transporter, we are talking about the concentration gradient. And this process, which could be against the charge gradient or mass gradient, and that is called as an active transporter. Now, in the active transporter, you have a class. One is a primary active transporter, another is a secondary active transporter. The primary active transporter is where one chemical involved, and whereas a secondary transporter is where it is a coupling the uphill transport of one substrate with the downhill transport of another. So one molecule enters, another molecule is coming out. It is again the same thing I have shown. If you have any doubt, we can discuss. Is it clear to you?
Yes, sir.
Okay, so let us move on. This further shows you, it depicts that how the concentration gradient can play a role. So C1 and C2 are the concentration. Suppose C1 is the outer side of the membrane, C2 is on the inner side of the membrane. There is a concentration gradient, and this molecule will start moving from high concentration to low concentration. And they will equilibrate, and at the equilibrium there will be no net flux. So you can see that there is an equilibrium in the second one. And here, once the concentration is equal, then C1 is equal to C2, and there is a no movement of the molecule. So here, what type of transport is this one?
Passive.
Unless
Passive.
this could be either, it is always passive transport, number one. Number two, if it is helped by the protein, they will be called as a facilitated transport. If it is not held by the protein, this will be called as a simple diffusion. Diffusion part you have already learned. Now, here we are going to talk about the before equilibrium. Suppose these are the charge, and at equilibrium, you will have this type of a charge distribution. So actually what it tells you that the charge gradient, so the gradient could be the mass gradient, the gradient could be a charge gradient. Now question comes that how the transporter, how the presence of a protein helps the movement of a molecule. So this is the one which is given. This is the one which this part shows you that how a molecule can move across the membrane. This is also giving you that how the molecule is moving with the help of a protein. So the first part, A, is a simple diffusion, and B is the facilitated diffusion. Now question comes that what facilitates, why facilitate, what is the mechanism, and how it is different. So if you look in the first case, what you have is you have a hydrated solute. And whenever we are talking about such mechanism, it is for hydrophilic molecule. So if I have a hydrophilic molecule, what will happen? It will be bound with the water molecule. And when there is a water molecule which is a binding, suppose I have a glucose molecule which has to enter and membrane. What will happen? Initially, outside the membrane, glucose will be surrounded by a lot of water molecules. So they are binding with the water molecule. And in a simple diffusion, the removal of the hydration cell, when they have to remove this, see, this has to be removed before they enter. When it is happening, it is a highly endergonic process. So what happens? The cost of energy, the energy of activation is quite high. So you look at this. Suppose I'm moving for the first A process, the activation energy involved is quite high. This is the activation energy, and that's why it is going. So simple diffusion requires high energy. And why it is free energy, high free energy is required? Because they have to remove the water ion. Now, take the example. Suppose I take a hydrophobic molecule. This hydrophobic molecule can move across the gradient because there you don't have a water bound to it, and that is why you don't need that to be removed. So if I go back to this slide, here it is very clearly illustrated or given that simple diffusion, it is for non-polar compound. And why it is non-polar compound? Because the non-polar compound, they don't have the water molecule bound to it, and that is why they can move across the gradient by simple diffusion. But if I look at a molecule which can have a binding with the water molecule, then the cost of energy is quite high, and because of that, what you require is that this energy should be lowered. And how this energy is lowered? Because you have a protein which is allowing this to move inside. What it does, that water molecule will bind with the protein. So your interaction is being taken care of by binding of the protein molecule. So actually what happens, that the protein binds with this the transporter, and then it allows the molecule. So by doing so, reduce the, instead of diffusion, you are having facilitated diffusion. And in this facilitated diffusion, the energy required is less, and that is the way transmembrane diffusion is held by the protein. And what are these proteins? These proteins are called as transporters. Now let us move on to ion channels. So you have seen I have named a couple of things. I said transporter, I talked about ion channel. So let us talk about what is the difference between ion channels and transporters. Ion channels, they speed the passage of inorganic ion across the membrane by mechanism which is a different from the transporter. So let us talk about what is the mechanism. So the mechanism involved where the ion channels, they are helping in the movement of inorganic ion. You will see that most of the ion, suppose this is the ion which has to move across the membrane. So you have this membrane because ions are the one which are having the ions, they are the one which are having the, the gate. If gate is closed, ion will not move. Once gate opens, the ion will move. Okay. Now, the similar kind of mechanism you will see in the transporter, but there is a slight difference, and we will talk about the difference. So most ion channels have a gate, and now question is, what regulates this gate? When it opens, when it closes? The gate is always opened through a signal. Now, suppose on the membrane, I have a receptor and a molecule comes, binds here, gives a signal, and this signal will open the gate. Is it clear to you? Can you, have you, understood the concept?
Yes, sir.
So in case of ion channel, the gate which is opening is regulated by a biological signal. Slowly, whenever we talk about one specific example, you will understand about this, this opening of the gate. And when the gate is open, ion moves across the membrane through the channel in a direction dictated by ion charges and electrochemical gradient. So the gate, if it is open, then the movement of ion will occur based on the electrochemical gradient. Ion channel typically shows some specificity for ion. What it means is that the signal which allows the gate to open for calcium ion, the same signal will not open the gate for sodium ion, or the signal which opens the gate for, say, the X type of ion will not allow the opening of gate for Y type of ion. So they are very specific signals, and that is the way there is a specificity of the ion channel. And second very important thing is that they are not saturable. What does it mean? That in case of a membrane transport, you will find that they are saturable. That means this molecule has to bind first with the transporter molecule, and then it is being transported. So if I say that if I keep on increasing the concentration of iron, and this iron binds with the protein molecule, and then it is transported, that means it becomes saturable. We'll talk about that in the next slide. Then channel stop either when the gating mechanism is closed. So here you may have a signal which says, okay, close the gate, or there is no longer electrochemical gradient providing the driving force. So this is opening only when there is a gradient which is created, number one, and number two, you have a signal. So here signal has opened the gate, and the ion, they move out. Now this is the both ways. What you are looking at is that gate, once it opens, it will allow across the gradient, means, sorry, it will be along the gradient. So if the concentration is high outside, low inside, from high concentration to low concentration, this moves, and this will stop only when the concentration becomes equal. And the opening and closing, this part will also be controlled by a signal. Signal can come, say gate, open gate, or signal can come and say close the gate. So these two things are important for ion channels. Now coming to the transport. If I look at the figure, you can say that you are opening the gate here, and in that case as well. So ion channels and transporter both have proteins, and they are, this is the mechanism by which it has been, explained. What you see is that the transporter transporters which binds to the substrate. Remember, in the ion channel, they do not bind with the substrate. So here, the first thing is that it will come, the channel will bind with first, and then, so what you are looking at, that this channel, what it does is that it binds over here, and then only it moves. So, and second thing is that they are saturable because the binding of the substrate occurs with your transporter molecule. So this is your transporter, and if the transporter binds with this, what will happen? If I have 100 such molecule and all the sites where the binding occurs is saturated, no more transport will occur. So unlike ion channel, the transporter, they are saturable and they will depend on the number of the transporter molecule or the concentration of the substrate to be transported. So that means they are saturable, and you can see this by doing the kinetics. It is the same thing like enzymes. Enzymes we have not discussed, but you will learn that in case of enzymes, the similar kind of a mechanism happens, that this means that if I follow the kinetics, it will kinetics. Transporters have a gate on either side of the membrane, and the two gates are never open at the same time. So look at this. Here, this gate is closed, this gate is open. This gate is open, this gate is closed. So what happens that here, that transporters or pumps, they alternate the opening of the gate. So first have what will happen? The transporter will allow the substrate molecule to bind to the transporter. Once it is bind, then what will happen? The gate may close, this might go out. And same way, the opening and closing of the gate is controlled by the binding of the solute molecule to the transporter. Remember, here the saturation occurs, whereas in case of ion pumps or ion channels, there is no saturation which is occurring. Here, why saturation occurs is because it binds to the transporter molecule. The other difference is that here, the one gate opens, another gate closes. But in that case, what you have is that the gate is open, another gate doesn't close ever. So once it is open, it will allow the molecule to move in. So this is a conceptual difference between ion channels and the transporters. Now looking at the kinetic mechanism, how this kinetic mechanism works. So here we have taken one example where we are going to talk about the glucose transporter, which is present in erythrocyte membrane. Remember that the glucose transport in the erythrocyte membrane may be different than the mechanism of glucose transport in other cells. So do not get confused. If you read in a book glucose, it is not universal. The transport of glucose may be different in bacteria, different in animal cell. Even within the animal cell, it can be different in different the cellular types. So let us talk about erythrocyte mediated passive transport. When I say passive transport, what is the idea coming in your mind? What does passive transport reflects? What do you understand by passive transport?
No involvement of energy.
Yes. So whenever we talk about passive transport, there's no involvement of energy, but you can have involvement of proteins or the transporter molecule. So energy yielding mechanism or metabolism in erythrocyte depends on the constant supply of glucose from the blood plasma, where the glucose concentration is maintained. So you have to maintain a particular concentration, approximately 5 millimolar concentration inside the erythrocyte cell. So when I'm talking erythrocyte, I think you understand it is a RBC, red blood cells, and in red blood cells, the energy requirement is there, and this energy requirement is met by the plasma where these RBC are suspended. They have glucose, which is pumping the glucose or which is going inside the cell. So glucose enters erythrocyte by a passive transport via a specific glucose transporter, and this name of this glucose transporter has been given as a GLUT1. Now GLUT1 could be different than GLUT2, 3, 4, 5, etc. So do not get confused by this. Now, question is that how much it is helping in the transport of the glucose molecule. You can see that the rate is about 50,000 times greater in those cases where it is held by the GLUT1. Suppose I look at the same transport across the membrane, because it is like, helping like a enzyme, or helping like a by lowering the by active the energy involvement. What you see is that this transport does exist. So the GLUT1 is not one which is causing a reaction to occur, but it is a catalyzing the reaction. So this is acting as a catalyst for the transport of glucose across the membrane. So the same phenomenon occurs. Suppose one glucose molecule is going inside the cell in the absence of GLUT1. What you will find is that 50,000 molecules of glucose, they go in the presence of this protein or GLUT1. So what you have learned is that protein is assisting the transport across the membrane, and the process of glucose transport can be described by analogy with the enzymatic reaction. As I said, if I talk about glucose, which acts as a substrate, that means when it is outside the membrane, when it is outside the erythrocyte cell, they are called as a substrate, and that is why we call it as a S out. And the product is when glucose enters inside the cell. So we call them as S in, or glucose inside, and the enzyme is replaced by T or transporter. So let us see the mechanism. It's very interesting. It can explain all types of transport. That's why I'm going to discuss about this. And you can find in many good books that how the transport mechanism has been defined. So what you have is that you have a substrate or a molecule which has to go inside the cell. They are present outside the cell. So suppose I have a membrane. Suppose this is a membrane. S is present over here. Now, when it has to enter, first thing what it will do is that it will bind with a transporter, and it is assumed that the transporter is present on the outer surface of the membrane. So first thing will be that S will bind with this transporter. So S plus T1. So there is a kinetics involved here. This is the constant, which is rate constant, which is given. So first thing will be S binds with the substrate, binds with the transporter, and then it becomes ST. So here I'm showing T1 because this is one type of a conformation of a transporter. So in one conformation, first conformation, substrate or glucose binds with the transporter, and still it is present on outside. So here it is S out, it is again S out, both of them they are present outside the membrane. Now once S binds with the T1, now what will happen? The conformation of T1 changes. It changes, shows some change in the structure, and that is why it can move from inside to outside. It helps the transport process. So this is to explain the mechanism. The real mechanism I'll tell you later on how it happens. But what will happen? That S1, which is outside the cell, they will move inside the cell. And there is again the constant involved, how it can move from outside to inside. So here, this is outside the membrane and this is inside the membrane. So this is outer side, so S out, it binds, becomes S out with T1, this is a bound form. Now the bound form moves in, and then this one gets dissociated, because once it goes inside, this one changes, your transporter changes its affinity with the substrate, and the substrate will be released. So here, what has happened? The T2 has released. Here T1 was one structure, T2 is another structure. Now this structure does not have enough affinity with the S1, and that S in, and that is why it releases, and S comes outside. So it is separated. Once it is separated, T2 again comes back in the same conformation. So what you have seen, this mechanism can be explained in the first step. What is this mechanism? What is happening here? It is happening binding. So first step is a binding. Second step here is a transport. And third step here is, what is the third step? It is a dissociation. And fourth step is recovery. So this is a cycle which is occurring, through which the glucose or the substrate molecule can first bind, then release, then again dissociate, and after dissociation, they are recovered. So there are 4 steps in which any transport mechanism can be explained. Tell me, have you understood this process?
Yes, sir.
Yes, sir.
Okay, any question I can address. Now, when I say that these transporter, they are saturable, what does it mean? Saturable means if I have extracellular concentration, I keep on increasing the concentration. And I look at the velocity of their transport. What you find is that they initially, they increase, and after a certain time of period, they become saturated. That means there is no more increase in the velocity of the transport across the membrane, even if your extracellular concentration is increased. That means why it is happening? Because the glucose molecule, they are binding with the transporter, and if the transporter gets saturated with the binding, no further transport occurs, because the first step in is the binding. So the initial rate of glucose entry into the erythrocyte, which is a V0, it is a V0, and it depends on initial concentration of glucose. So initial concentration, you are increasing, it is increasing here. So initially transport increases with the increasing concentration of glucose outside the cell. But as soon as you keep on increasing here, something has happened where it gets saturated and there is no more increase in velocity of glucose transport. Now I will talk about Vmax, Vmax half, and Kt in a different reference when we talk about the enzyme. Actually, Vmax refers to maximum velocity. And if I take out half of the Vmax, at this half of Vmax, the concentration of glucose is referred as a constant. In case of enzyme, we refer as a Michaelis constant or Km. Here we say Kt. Kt refers to transport constant, nothing else. Now, this expression, I will give you expression how this can be expressed. If it is converted into the single line expression, it can make 1 by V0 versus 1 by substrate concentration. So it becomes a straight line. So all these things we will talk about this kinetics when we talk about the enzymes. But here it gives you information that the transport process are saturable. And why it is saturated? Because the solute molecule, they keep them, all of them, all the active site or the binding site are filled, and that is why it happens. Now, can you tell me this graph, what does it tell you? This tells you it's saturated. That means that binding of the solute molecule to the transporter is essential step. If it is not binding, then there will be no saturation, as you have seen in case of ion channels. So ion channel, there is no binding, and that is why there is no saturation. But the transporter, because of the binding, because there may be a concentration where all the binding sites, they are full, and that is why no more transport occurs. Now coming to the further explanation, there are substrate molecule which can bind and they can be transported. Here, the transporter allows only one molecule to be transported. So suppose glucose has to be transported, just glucose will bind with the protein and then they will move on. On the other hand, what happens? Glucose binds, but at the same time, the transporter has a binding affinity for another solute molecule as well, or any ion as well, and both of them, they will move together. So in the same port, the same protein is helping two molecules, S1 and S2, to move inside. So that is why it is called. That means both of them, they are moving in the same direction, and one is helping in the transport of other. So it is a symbiotic process where both of them, they are moving in one direction. Now it could be different. It is possible that there are two molecules, S1 and S2, both binds with the same protein, but they are moving in a different direction. That means suppose I have a potassium ion or a sodium ion, which moves from this side to this side, and after moving, it creates a gradient, and that gradient helps the another molecule to come out. So this type of transporter are called as an antiport because the direction of the movement is different. But both of them, they are co-transport. That means they need one molecule to help the other in the transport process. Now, I will try to explain what is a primary active transport and secondary active transport, which we have talked about in the first one. So what you have is that in primary active transport, the energy released by ATP hydrolysis. So you see that this is a transporter, ATP is hydrolyzing, it is giving ADP plus PI, and this is the one which helps S1 to move outside the cell. So movement is against an electrochemical gradient. So what you are looking at is that here S1 is only one molecule. Here it is so many molecules. So what should happen? Actually, if it is not energy dependent process, then solute molecule should move from outside to inside. But here it is happening reverse. Why it is happening reverse? Because it is energy which is taking care of pumping out of the solute molecule. So in the primary transport, what you have learned is that it is the concentration gradient which is important. And so this is the way it happens in the primary active transport. What will happen in the secondary active transport? Secondary active transport, what you learn is that S1 first move outside with the help of energy. And when S move outside, what will happen? This helps the S2 to move inside. So both of them, so energy is coming from here. It helps the S1 to move outside. And because there is again a, this has pumped out. Now S1 concentration will change outside, and this will allow S2 to move in. So this kind of a transporter you will learn where sodium potassium transport occurs. Sodium ions, 3 sodium ion which is present at one time, 3 sodium ion, any animal cell, they move outside. And this happens through a transporter, which is known as ATP, sodium potassium transporter, helped by ATP. The C sodium ion will move outside. And this moving outside will, both the time it has been explained that this change in the structure of this transporter occurs. Now, transporter structure has occurred. They allow the sodium ion to move in. This helps the potassium ion to move from outside to inside. So 2 potassium ion will move inside. So now what you are getting is that if I say at a given time 3 sodium ion comes in, sorry, 3 sodium ion goes out, 2 potassium ion comes in. So what kind of a transport it is? It is a secondary active transport, as well as what will be the charge gradient inside, how the membrane potential will change. Inside it will be negative or positive.
Negative.
It should be negative. So it is the helping in maintaining the negative potential inside the cell. And once you are having a charge gradient which is created, so this transport is also known as electrogenic transport. That means it is creating a charge gradient. But on the other hand, if I say one potassium ion goes out and one proton comes in, so that will be charge neutral. So that is another way by which you can differentiate between different transporter. So what I'm talking about here is that primary active transport is establishing the gradient, and then it provides the energy to drive the co-transporter, that is S2 molecule, to enter in. So this kind of the transport is known as a secondary active transport. Now there is a very important thing which we'll discuss, and probably today we may not have enough time, but just to give you an idea that from where this energy comes. So most of the time, the amount of energy needed for the transport of a solute against the gradient can be calculated. You can find out how much is the concentration gradient and how much is the charge gradient, and based on that, you can calculate the free energy change. And this equation is known to you from very beginning, that you can calculate the delta G and tell delta G is a positive for any kind of a calculation where transport is occurring. What type of transport occurs? Will it be active or passive?
Active.
So when delta G is a positive, that means you have active transport. If delta G is negative, it is a passive transport. It is very clear. Now you can calculate the delta G value. Now it is a general equation. All of you know that P is a product, S is a substrate. So what will be a product and substrate in case of a transport? A molecule which is present outside the membrane, that is the substrate. And when it enters inside the cell, it becomes a product. So always we give product by substrate, which is given by C2 by C1. Okay. Here I have given this equation. Now I hope that you understand what is delta Z prime, delta Z0 prime. It is the standard free energy change, and it is simply the when this reaction is a simply transport of a solute from region where its concentration is C1 to region where it is a concentration is C2. No bonds are made or broken, and delta Z0 prime is 0. So the free energy change can be calculated. This G is delta G, or here it is written G T. Implies for transport or transporter. So it can be given by RT ln C2 by C1. And suppose there is a difference of a tenfold in the concentration, you can calculate by putting the similar things, values of R, T, and C2 by C1, and you can calculate the delta GT value, and that tells you whether the transport process is active process or it is a passive process. I will stop at this point. We have a couple of more slides which we'll discuss in the later stage, and you will also learn that not only the mass gradient, which is important, there is also contribution of a charge. So we will calculate and learn how the charge gradient plays an important role. So I'm stopping it at this point. You can, let me know if you want to understand anything out of this. So if it is everything is clear, it is fine. Otherwise, let me know what you need to, elaborate and give you, explain you. Is it clear to you?
Yes, sir.
Most of the concept you might have learned in one or the other classes. Even in the chemistry, it is being taught. Physics, it is being taught. So here we are translating those concepts for the biological phenomenon. So it's very clear that delta G can be calculated, and you can, based on that, you can find out whether the process is active process or passive process. Any questions? Anyone?
Sir, I have a question.
Right.
Sir, between active and passive transport, which one is more spontaneous in nature?
See, both, whether it is active or passive, none of them will be spontaneous. Everything has to be based on the concentration gradient. And if you find out difference between the general diffusion and passive diffusion, I can say that the normal diffusion process will be held by the presence of a protein, and that will be more, spontaneous. That will be, much faster. So if you look at the rate will be faster when it is facilitated by the protein. Between active and passive process, you cannot differentiate between active and passive process in terms of their spontaneity, because uh, the uh, the passive process is also held by the protein, an active process. So the difference between these two is because of the energy. Now I can tell you how actually experimentally you can do it. So that part we'll discuss in the class. But think about the reason. How, if I have to prove in a cell, it is very difficult to find out what is the delta G value. Because at a given time, getting the concentration across the membrane is quite difficult because it's a continuous process. So now question is that quickly I want to do an experiment and find out whether in E. coli the glucose is transported across the membrane by active process or by a passive process. How to find out that? So we'll discuss this part. Any other question? If not, then I will sign off and will continue with the transport across the membrane. Thank you
Thank
Thank
you.
you,
guys.
sir.
So, could you tell us a
Yeah.
little about the minor, the pattern exactly?
from the lectures, most of the concept, but I will try to give you logical questions, and it will be short answer type of question, which is a common, and you will have a number of questions because it is more or less, it is not monitored examination. That means you have access to everything, and because of that you will have large number of questions which will be short answer type, and it could be logical as well based on whatever has been taught. So think about the logical question. You should have the molecular logic, and based on the molecular logic most of the questions will be there. Okay.
So also, what will be the platform for the topic?
See, platform, I'll let you know within a day or two. I will write in your, so first thing is that you will get the question. It could be given by your, either through the Moodle or through the different modes. We are just debating, and you will see that where you can, work on. That will not be a problem because I will inform you in advance, and I will be using the most common platform which is being used for the other exam. Any more
Thank
question? Okay then, thank you. I'm signing off. Bye.